Hi everyone, welcome to BMI 4302 Virology. This course is brought to you by the Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University of Putra, Malaysia. I'm Dr. Benicia. So in previous lectures, we have talked about Baltimore classifications of viruses, which is based on how the protein has been synthesized from the genome of the virus. So there are 7 groups of them. We have RNA and DNA virus, where in the RNA virus, we can separate them into single-stranded and double-stranded. And in single-stranded, we have single-stranded RNA that is positive orientation or positive strain. We also have single-stranded that is negative strain. Of course, we also have double-stranded RNA. A special case for this RNA is that there is a single-stranded RNA that undergo reverse transcriptions to produce their viral protein and their viral genome, which is the Picornaviridae or the HIV virus. Then we also have double-stranded DNA and single-stranded DNA. So in double-stranded DNA, we also have another group of viruses that undergo reverse transcriptions as part of their life cycle. So in total, there are 7 groups of the viruses. So because the virus, there are so many of viruses in the world, so it's not possible for us to cover every single virus during this short period of time. So I have selected a few family of the viruses for this course. For example, for single-stranded positive RNA, we have selected the one that without envelope. We chose Picornaviridae. Then the one with envelope, we chose Flaviviridae. Then in negative strain, we have chosen the Paramyxoviridae, Automyxoviridae. And to make it more interesting, I've included the Retroviridae as part of the course. And for double-stranded, I've chosen herpes viridae as our study in this case, and we have chosen Picornaviridae as our course. So what I believe is once you understand one of the virus in detail, you'll be able to apply the same concept throughout the rest of the virus. So that's the reason why we have chosen some of these viruses family as a topic to be focused on. Alright, so let's start with our first topic, which is the naked positive-stranded RNA virus. So basically the outline of this course, what we are going to cover is we'll be covering some give you a brief introductions of how what is the virus, positive-stranded RNA virus look like, and what is the characteristic of this virus. And of course, then we'll move on to selective non-enveloped or naked RNA virus, which we have chosen Picornaviridae. Then we have chosen the enveloped virus, which is the Flaviviridae. Then what we're going to do is we're going to study in detail their life cycle of a selected virus. For Picornaviridae, we have selected poliovirus, and for Flaviviridae, we have chosen dengue virus. So that once we understand the life cycle in detail, and you'll be able to apply this for the rest of the virus, as I mentioned earlier. So, positive-stranded RNA virus, what does it mean by positive-stranded RNA virus? It means that their genome is single-stranded. So we mentioned that the RNA or DNA, they have single-stranded and double-stranded. When we say a single-stranded or we say double-stranded, it has a 5' to 3' as well as a 3' to 5' that come together to form a double-stranded. So when this double-stranded is being separated, the one 5' to 3', that will be the positive strain. The one 3' to 5', that will be the negative strain. So I hope you understand what it means by positive strain and negative strain. So the positive strain RNA have a single-stranded, of course, if it is just a positive polarity or positive strain, so it's just a single-stranded. Alright, and we also mentioned in the previous lectures in order for a protein to be produced, it must go through a transcription, which is from DNA to RNA, a process called transcriptions. And once the mRNA is being produced, this mRNA will then be translated into protein. Okay, so the mRNA is the messenger RNA, which is very important. And we also talk about like the orientation of the messenger RNA, which is also a 5' to 3'. So in here, what we can see is that the genome of this RNA virus is 5' to 3', means that the genome of the RNA, Positive-Strand RNA virus can be used as a functional mRNA, means that the genome can be directly used for the productions of protein. Okay, so now it raises the questions. Does it mean that the Positive-Strand RNA virus has all the characteristics of the mRNA? And we also talk about what is the characteristic of mRNA. The mRNA itself must have a cap, which is 7-methylguanosine, and it has poly-A tail, and this mRNA must be spliced in the nucleus before it's being transported out from the nucleus into the cytoplasm for translation. Whereas for this virus, the RNA, does it contains 7-methylguanosine? Does it contain poly-A tail? Has it been spliced? We don't know. We'll talk about it later. Alright? So then this group of virus contains a large group of viruses, because this is quite, the RNA virus is a very large group and cause a lot of human diseases such as encephalitis. So encephalitis means inflammations of the brain, hepatitis, inflammations of the liver, it causes yellow fever, dengue fever, poliomyelitis, as well as common cold. Alright, so for the purpose of the course, we focus on 6 families that causes human diseases. The reason why we focus on human diseases, of course you can later on use this technique to study other plant diseases, virus, bacterial diseases, animal diseases as well. But the reason why I chose human diseases is because it's close to us, so you can relate it easier, then you'll be able to understand the virus more. So these are the 6 families that cause human diseases. So we have Picornaviridae, which is has a genome size of 7,500 base. Now be careful because it's a single-stranded, so we're going to call it a base pair. So 7,500 bases, where the genome, the virus cause poliovirus, human rhinovirus, hepatitis A virus. So human rhinovirus is the virus that causes the common cold, that we get it quite often. Also, we have hepatitis A virus that causes the liver inflammations. Then we have Positive-Strand 7,500 bases as well. In this group, we have a norovirus, a virus that causes the gastrointestinal or gastroenteritis, right? Gastroenteritis. Then we have Positive-Strand. Size of the genome is about 6,800 to 7,900. Okay, the representative of the virus is a human astrovirus that causes gastroenteritis as well. Next one, what we have is Togaviridae. Now, Togaviridae have a genome size of 11,600 almost or more. So the disease that caused by this group of viruses are Rose River viruses, which is a mosquito-borne virus. We have Western equine encephalitis, Venezuela equine encephalitis, as well as Eastern equine encephalitis. So these are the mosquito-borne diseases. Okay, that when it was identified in the horses, and then later found that the mosquito also cause diseases in the virus also cause diseases in humans. and it's quite a serious one, encephalitis. Right? Then we also have Mayaro virus, is also a mosquito-borne virus, and then rubella, German measles. The next one is Flaviviridae. So it has a size of 9,500 to 12,500. The very famous disease that caused by this virus is the dengue, yellow fever, Japanese encephalitis, Murray Valley encephalitis, tick-borne encephalitis, as well as hepatitis C. Now, I would like you to notice that even though hepatitis A and hepatitis C cause inflammations of the liver, they belong to the different family. Okay, so don't assume all the diseases that cause at certain organs come from the same family, right? So we have hepatitis A that belongs to Picornaviridae, where hepatitis C belongs to Flaviviridae. We also have hepatitis B that belongs to Picornaviridae, a totally 3 different families. Okay, and lastly we have Coronaviridae, have a size of the genome of 20,000 to 30,000. Now, so the common disease that common virus that cause the disease in human is human coronavirus. So there are few type of coronavirus. The general one that is called common cold, but we also have a more serious coronavirus that also caused like SARS in 2003. Then we have MERS, Middle Eastern Respiratory Syndrome, MERS coronavirus, and in 2019 we have COVID-19 that causes a pandemic around the world. So what we notice here is that the small signs here also indicate that they might not be necessarily true, but in this case what we can see is like 7,500, 7,000 to 8,000. So this group of viruses are naked viruses, and this group of viruses are the enveloped virus. So the one that we are going to focus on is Picornaviridae. So family of Picornaviridae, from the name Pico. So we know centi, which is -3, 10 to the power of -3. We have micro, 10 to the power of -6, nano, 10 to the power of 9, and then pico is 10 to the power of -12. So what it means is that it's really small. Okay, pico, small, and it is RNA containing virus. That's why this group of family is called Picornaviridae. So a little bit of tips. Sometimes for us, if you remember it as Picornaviridae, and when you try to write it, you might mistaken spell it as Picornaviridae. So during my time of being a student, so how I remember is Picornaviridae. This is how I remember it, so that I won't miss the R here. Right? So, but it's up to you how you want to memorize how you spell the family. So basically, it's pronounced as Picornaviridae. Right? And the next thing is like when I was students, there are generally only 9 general, and now it has increased to 47 general, and only 5 general that cause human pathogens, but now we have 8 general that cause human pathogens. So there are 2 explanations here. One is like we know more about science when we understand the virus more, and we have better bioinformatics tools that which allow us to reclassify our viruses, rather than it used to be grouped under the same name because they cause the same disease. And now, because after identify the genome, the sequence of the amino acid and nucleotide, then we can separate the viruses into different genera more accurately. So science is advancing, and especially virology. So maybe when in the future, when you start looking back at Picornaviridae, here might become 100, here might become 40 human pathogens, who knows. Alright, so what we're going to see is like the 8 general that cause human disease. We have enterovirus, paracorvirus, cardiovirus, kubo, hepato, and this is what I learned when I was student. And now we have Cosa virus, Salivirus, and Rosa virus. So in total, there are 8 general. And in enterovirus, we have polio, ecovirus, Coxsackie, and enterovirus, and rhinovirus. So most of the disease, most of the virus transmitted through oral and fecal contact. So for a student, sometimes they find it very difficult to imagine how is it being transmitted through oral fecal. Well, in country that is underdeveloped, where the sewage system is not really well established, so you tend to have the water and the sewage getting mixed or accidentally pour into the drinking water. So in the end, if someone is infected or by the virus, that will then be transmitted to the water, and if you drink it, then you will have you will have the disease, and this is how it's being transmitted. So personal hygiene is very important, especially after you have gone to the toilet. Make sure that you always clean your hand, wash your hand with soap, and make sure it's free from any contaminants. But also we have rhinovirus that spread through aerosol. This is the one that causes common cold, right? Rhinovirus. And also here, what I would like to introduce to you is some definitions, some terms. Okay, meningitis. What does it mean by meningitis? So you have encephalitis, means that inflammations of the brain. So meningitis means inflammations of the meninges membrane. So our scalp, our scalp, underneath your scalp is your skull. So your skull and your brain, in between them there is a layer of membrane, and this layer of membrane act as the protections to protect your brain, and this membrane is called meninges. Right? So when that layer of membrane is inflamed, it's called meningitis. Okay? And what does it mean by aseptic? So inflammations of this layer of membrane can be caused by bacteria and can be caused by virus. So when it is caused by virus, it's called aseptic meningitis. Right? So myocarditis means that inflammations of the heart muscles. Right? Meningitis. Conjunctivitis, inflammations of the conjunctiva. So this conjunctiva is actually the layer underneath your eyelid. Alright, this is the membrane underneath your eyelid, and I guess that's it. So you should understand all this term. Okay, so we mentioned that the classifications of the virus is based on the nucleotide and amino acid sequence. So I'm not sure whether you have seen this before. This is called the phylogenetic tree. So what does it mean by phylogenetic tree? So we know the nucleotide sequence, and when you know the nucleotide sequence, you can convert it into amino acid, and I'm sure you know how to do that. So basically, what the scientists are trying to do is try to identify all these viruses and see whether they are related to each other. And what is being done here is they use the amino acid sequence of these viruses, put them together, and try to align them and see how many of these viruses are closely related. So you will learn how to build this phylogenetic tree during a bioinformatics course. Alright, so what we can see here is based on the amino acid sequence, you can safely classify the viruses into groups. For example, hepatitis A, and then you have here Coxsackie, enterovirus, poliovirus. They are all under the same general, which is enterovirus. then you have outdoor, cardio, and these are the viruses that are quite close to related to each other, and that's why they are fall under the same branch. So I'm not going to discuss this in detail, but basically what I'm trying to say is once you understand the phylogenetic tree, you'll be able to see how the classifications of virus being done, and that's the reason why we're getting more and more general in the future. Alright, so the one that we're going to focus on is poliovirus. So poliovirus fall under the family of Picornaviridae. Of course, we have been talking about Picornaviridae, and above the family is orders. It fall under the Picornaviridae. So during my time, there's only one order, and now we have so many orders for the virus. And the genus Enterovirus. Okay, so please notice that when you put the scientific writing for genus, it must be italics. And you notice that there's also another Enterovirus here. So what it means, if you see a genus that is being like italicized, which is the case here, Enterovirus, it represents this virus, this virus, this virus, and this virus. But if it is not italicized, so it will be this virus. Okay? So in virology, it's very common for people to try to describe a virus by starting saying that, poliovirus is a member of the order Picornaviridae, family of Picornaviridae, genus of Enterovirus. This is a common way of us writing the introductions of how of a virus. So over here, what you see is a TEM, transmission electron microscope photograph of the poliovirus, and what we can see here is all these viruses are having an icosahedral structures. All right, and this is a more detail of the SEM, okay, scanning electron microscope, so you can see a more detail of the surface of the virus. So this virus is being transmitted through oral fecal route. What it means is that when you consume water that it has been contaminated by this virus, so the virus will go through your mouth and start infecting your gastrointestinal tract. So for example, your throat or your intestine. So once the virus get access to your cell and go into your lymphatic system, then it will start spreading through your blood throughout your body. This is what we call a viremia. The presence of virus in your blood, viremia. Alright, so poliovirus have a specific target cell that they infect. The cell that they infect is actually the nerve cell. So when they attack the nerve cell, and we know when virus go into a cell and they replicate and eventually they will kill the cell. So what happened is they kill the nerve cell. And we know that the nerve cell is very important in our body, and once the nerve cell is being destroyed or disrupted, it's very difficult to repair them. Alright, so poliovirus infect the nerve cell and it causes death of motor neuron in the spinal cord. So this is a disease that caused by the poliovirus called poliomyelitis. So when the spinal cord, the motor neuron in the spinal cord is being injured or being killed, what happens is your muscle will no longer receive the signal from your brain. So meaning that your hands or your legs will no longer be able to contract or release. So what you can see here is when you are not using a muscle for too long, it will start to shrink, the muscle becomes smaller, and eventually, and this is not moving at all. And what you can see here are some of the people who are suffering from poliomyelitis. So let's look at the structures of this virus. Now, the structure of virus, even though it looks pretty here, but in actual fact is colorless, so you can't see any color at all. Alright, so the structure of the virus is made up of 4 major proteins called viral protein 1, VP1, VP2, VP3, and VP4. So these form the protomer of the virus. But if you prefer colors, okay, VP1 will be the one in blue, VP2 will be the one in green, VP3 will be the one in red. We also have VP4, which is hidden at the back of the capsid. So here is yellow color here. So this VP4 is hidden and protected underneath this VP1, 2, and 3. So this form the protomers, and when 5 protomers that group together, they become the pentamer, and eventually they form the capsid of the virus, and what we can see from here. Now, I want you to notice that, like, if you look at the structure of this virus, you will notice that the virus is not a smooth, the icosahedral is not a smooth structures, and you can see they are up and down, which form a canyon, alright, a groove, and this is the spike up, and so on and so forth. So these structures on the surface of virus is very important, as it behaves as the site where the receptor binds to. Okay? So when we cut the virus into half, what you can see is inside the core of the virus, this is where it lies the genome of the virus. So let's look in detail of how the genome looks like. Now, just now we mentioned VP1, VP2, VP3, and VP4. Now, this is actually the part of the genome on the virus that coded for the capsid. So the genome basically can be separated into P1, which coded for the capsid, or we call it the structural protein. Then we also have P2 and P3, which coded for non-structural protein. So what are the proteins that being coded here? Some, for example, the enzymes, polymerase, or some protein that is important for the virus, replications. Alright, so remember we also mentioned that the virus is positive-strand, which has a 5' end to the 3' end. Okay, so you have region of the genome that coded for protein. That's why this is area we call the coding region. However, the genome of the virus also have non-coding regions, meaning that the region of the RNA that is not being translated into a protein. So for example here, this is the region of RNA that is not being translated into any protein. So this is the area we call untranslated region. Untranslated region. And because it's located at the 5' end, so we put 5' untranslated region. 5' untranslated regions. What about here? We also have region that is not being translated into the protein, so we call it a 3' untranslated region. Sometimes there are different terminology, but they all carry the same meaning. Sometimes you can see NTR mean non-translated regions, and so on and so forth. Alright, so the other things that I would like you to notice is that, we all learn that the genome, the DNA should only contain nucleotide, or RNA should only contain nucleotide, and it shouldn't contain any protein that covalently bonds to it. So virus is very special in such a way that for poliovirus, they actually have a protein that covalently bond to the genome of the virus, such as here we have VPG, stand for virus protein genome link. Alright, so what it means here is that this protein is bound covalently to the RNA of the virus. Alright, so virus is very special and very interesting. Alright, so basically this is how the viral genome look like, and this is another diagram to show you, and yeah, so coding domain or coding regions and non-coding region and so on and so forth. So remember we say when we try to classify the viruses accordingly. Now this is how it looks like. When we put all the virus together, we notice that the general, okay, within this enterovirus, what we can see is that they have P1, P2, and P3. The arrangement of the genome is always similar. Okay, maybe not exactly the same, but it's quite similar. For example, here now the difference is that here we have L, but this one doesn't have L, and so on and so forth. But the arrangement is almost the same. We have P1, P2, P3, and then you have the capsid being formed here and non-structural being formed, located on the second half of the virus genome. Alright, so that's the end for the first part, where we basically what we have talked about in these sections is I give you some introductions of what positive RNA virus is, and how does it look like, the characteristics, and then the classifications. For example, the Picornaviridae or other viral family. So I hope you enjoy it. That's it. Thank you.